project is based on the premise that there are some limitations to the existing methods of the connectivity mapping paradigm as far as selecting the query gene signatures, that there's not really an optimized way to do it, and that the size that's selected and other properties dramatically affect the outcome, and they kind of compare different analyses and show that the selection of those really affects the outcomes. So, their premise is that this new method, the Dr. Insight, it's a acronym for Drug Repurposing, Integration, and Systematic Investigation of Genomic High Throughput Data. It doesn't involve using signatures, but it does allow the analysis of gene expression for repurposing, drug repurposing, and so basically they just introduced this new statistical model to combine the disease and drug data. And yeah, so anyway, the package is available on CRAN, and the paper, if you're interested, they have a supplementary table that compares their method against other connectivity mapping-based drug repurposing methods, which is pretty helpful to see how they all compare as far as their characteristics. And that, yeah, so that's the first one. And then the second is our package called Slinky, and it basically allows streamlining of the processing of L1000 data. It is available on Bioconductor, and it was created to integrate differential gene expression and gene signature analysis with the L1000 platform. If you go down to Figure 1, it shows an example kind of workflow for being able to combine the geo expression data and metadata and the clue metadata with the L1000, that part A. And so it's a pretty short paper, pretty simple package. They cite Zichen's L1000 FWD paper for how to process the control samples. So it looks like I think they use his method within their package. And so anyway, it's supposed to provide just a streamlined interface to the L1000 data and associated metadata. So it should help with simplifying processing. And that's it.
Website. So this is an AI-based automated way to create logos. So if you go on create logo. So for example, if you type in GeneShot, I think it's just like a funny result. I think you can just go on. You type a tagline. So you can give it keywords, but I don't think, you can you don't even need to do it. You need to do it. Oh, okay, just type in I don't know, gene. Okay. And then if you let's say vibrant, so you can specify a color scheme, and then it kind of builds a whole lot of suggestions of logos. And it's shockingly close to what we actually use. So if you start scrolling over, so it'll give you a bunch of ideas. Obviously, if you, and then so this is just I don't know if people know this. So this is a company that was I think founded in 2009, and it's by I don't know Stanford professors or something that created this. And it's interesting because it basically allows RNA-seq and in general sequence alignment in the cloud. And what's kind of interesting is that I feel in terms of the abilities, it's not. I don't think it's actually that advanced. But the valuation of this company, I think they collected over $100 million in seed funding.
So this is a new sub-package of TensorFlow, called TensorFlow Hub. So it's a collection of parts of neural network modules that can be easily reused, and it's also a platform to share those modules or layers. So on the example on the right, it shows you can easily reuse an embedding layer from this hub. It's just getting the URL, and then you can this layer to encode any languages. And if you click the blue button, there are a few more reusable modules that you can adapt to your device. Yeah. So this is a similar effort to Jupyter Notebook, but I think they're really using JavaScript to do the scientific computing and generate those notebook style reports. So if you click on any of the examples,
It's
then
like the most popular, like,
yeah, so that's the interactive notebook you can explore. And you can also click on the explore button on the top right to see the source code of this notebook. So also
It's just Markdown. It's pretty cool.
I can use both Markdown and JavaScript, and then all the analysis can also be done in JavaScript.
Yeah, anyway, this first one is just a resource for like rare cardiac diseases, and basically it has if you like click a disease and the annotated diseases, it just shows some annotations. And what I like is if you go up, down. Yeah, they have like the list of the symptoms or the phenotypes, and then they have also the abundance or how frequent that phenotype exists for people with that disease, and then they also have like the degree frequency or how unique that phenotype is to a specific disease. So I think that's just interesting and cool if you're like trying to diagnose a rare disease or something. This is just a visual of like papers from arXiv. Yeah, so their connection is based on who cited you and who you cited. So so that's how they did the nearness or something. So and then if you click a circle, the larger the circle, it means you have more citations or something like that. You get the metadata or something.
Yeah, 3.
Then you may be able to get like related papers if you're like, depending on your interest.
Oh, that's pretty cool.
Sure. So this one was published in Bioinformatics recently. The website, if you try to upload your own PDF, actually, go to browse. Yeah. So it'll actually just upload a sample PDF, and I couldn't figure out how to upload my own. But the reason I wanted to show this was because figures, like I know GeneShot takes the abstracts, which would give you relevant associations. That figure captions, I think, would also give really relevant associations because they essentially summarize the findings of the paper. So these people mined them from PDFs, so I imagine it would be easier to do from. So the other website I wanted to show is this My Heart Counts. It's an iPhone application. Over 50,000 people used it and have consented to have their data used. And the reason I wanted to show this website is because this week they released 50,000 people's data on Synapse. You can go to Synapse. I downloaded the metadata. They have motion tracking data. They have surveys, including like, are you depressed? Are you anxious? Are you happy? Do you have a sense of well-being? They have all this data. They also have sleep data for these people and their workout data. If they linked their workout app to this app. And all you have to do is agree with the terms and answer a few ethics questions surrounding your use of the data in order to get it. So it's all on Synapse now.
So this is pretty exciting. So I don't know if you guys know Shan Zhao from Ravi's lab. So he started a company together with some other person from California that is more of an entrepreneur. And the idea of the company is to have like a 23andMe for pets. But basically they give you like an adhesive tape where you can pull hair from your pet, and then they're gonna extract the DNA from the roots of the hair, and then sequence it, and then give you a profile of your pet. And that also use that for diseases, like you potentially can say that the pet has some type of a genetic disease or something, and it can be used for research. And they're going to be on Shark Tank on April 28th. And based on the way the site looks, it looks like they got the funding from Shark Tank because it looks really professional. And they have an advisory board, and it looks like it's taking off.
So this first is kind of like a repository of different biological data sets which have all been converted to the RDF format, kind of related to Daniel's talk a couple weeks ago. And I think it's pretty interesting. It seems similar to the Enricher effort in terms of converting to GPT, but just in this case they have a different, so if you click there on statistics, it just breaks down for different data sets they downloaded, and it's a combination of proteomics, genomics, transcriptomics, any sort of gene-gene relationship or gene-term relationship, and they have billions of these associations. Data sets, there's also more detailed description of dates that have been where each of these things is in the process, what kind of information it contains, and also on the left at the bottom, we have the Sparkle query engine, so it supports programmatic querying of all the data.
So this is just like a visualization of success over time for different scientists. So basically they're just using like citation data to look at every person's career. And they found that it's really random when someone becomes successful. I think I should skip. But basically you can like filter by like number of papers, number of citations. It's not like super useful. I wish you could like select things like based on either like name or something like that. So it's kind of just like jumbled up, but it'd be pretty cool if you could do a little bit more with it.
So what are those peaks?
So each one, so you click on one. So that this is for one person, and it's their publications over time throughout their career. And this was published in Amsterdam Genetics recently. Basically just classifies PubMed articles. So you can give it a list of PubMed IDs that you're interested in, and then PubMed IDs you're not interested in, and then a list of PubMed IDs you want to classify. And it trains a bunch of different classifiers, picks the best one, and then basically tells you which ones are relevant that you want to read. So that would be useful like if you're writing a paper and you have this like the citations that you're using in your paper, maybe like come up with.
This is a EVI's project, EV knowledge base, and it just accumulates community driven data for protein mutation, for protein ligand interaction, for variants, for functional and phenotypical integration, and so I'll go backwards. This I found, it was pointed out to me by Michel Demontier, and this is kind of a format for compressing RDF files. So typically these RDF files are this like XML, and it's gross, and then this HTT is just a binary format of RDF that can be queried pretty quickly, and it's part of the W3C standard, so it's just something that can be used in the future. Then this Big Data Europe thing is actually pretty interesting. When Konstantin mentioned to us about kind of using a more scalable compute engine for the Signature Commons backend, I
had actually looked into this before, and I found this, and then I refound this again. And it turns out this is actually an open source platform, and frankly, I think it really should be what Gen 3 is trying to do. There's a whole part in here about semantic data as well. And so essentially, they talk about something that I was kind of like trying to do, which is that you can have your completely unstructured data in your so-called data lake, they call it in the big data world, and then you can write wrappers which turn that unstructured data into structured RDF, and then you can actually perform knowledge graph type queries on it. And then this is along the same vein. This is like, okay, an ecosystem of user-facing microservices supported by semantic models. It turns out behind the scenes, this is also utilizing the Big Data Europe platform, but they extend it furthermore and really like talk about utilizing semantic technologies embedded in microservices.